Hi there and welcome to our first lesson in our B4 topic. We're looking today at It's All Green, so we're going to be looking mainly at the biology of plants. We'll be looking at a little bit of ecology through this as well, and a tiny little bit about humans as well. Now, what we are going to look at in this lesson on sampling and ecology is how we can actually catch animals and look at how we can find out how much there is of each of them in a particular habitat. Anyway, I'll go on to our objectives now. I'll see you at the end. Bye. Okay, so here are our objectives for this lesson on sampling and ecology. So by the end of the lesson you should know and understand a variety of techniques used to sample populations of organisms and how to estimate the population and display that data. Okay, so first of all we're going to look at what a population actually is and how we can actually sample them. So a population is a total number of individuals of the same species in a certain area. So for example, it could be the number of mice in a field, the number of woodlice under a rock, the amount of grass in a garden. It can be any of those things. Now, the size of the distribution of those populations can be measured using various techniques, and that is called sampling. Now, when you are sampling, what you need to do is first and foremost you need to get a very big sample. Now, a big sample will mean that you can get good and reliable estimates. So the bigger the sample, the more reliable the estimate. The second thing to do is make sure that you sample randomly. The more random it is, the more representative of the population it will actually be. Okay, so what we're going to do next is have a look at the different ways in which we can collect organisms to see what the sample will look like. Now, the first sampling method we're going to look at is called a puta. Now, a puta is a piece of equipment like this. Now, it's got a plastic see-through container here with a lid, and it's got a tube going in and then a tube going out. Now, the green tube here has a wire mesh at the end. Okay, so what you do is you suck the green tube, and that creates a vacuum in here, and you can suck up the organisms into the pot. Now, the wire mesh is to prevent those organisms, those insects, from getting sucked up into your mouth. Now, what this method will do is it will give you an idea of the organisms or the species that are actually present in the habitat, but it won't give you an idea on population size. Okay, and when you do this, you do have to be systematic with your sampling method. Okay, so maintaining that randomness, but you do have to be systematic with that randomness. Now, the next method we look at is sweep nets. Now, the sweep net is like a net, but you can see here it's a lot more smaller holes, a lot denser lining to allow you to collect those organisms. Now, this is a much better use in shrubs and woodland. Again, it's difficult to get an idea of relative size of the population, but you can get an idea as to what population there is. Now, a pitfall trap is essentially what it says on the tin. It's a trap in a pit that allows organisms to fall into it. Now, I'm going to show you this diagram or this picture of one here, and you can see that all they've done is they put a paper cup, they've dug a hole, put the paper cup in, and it allows organisms to fall into that cup. Now, you can leave that for a certain period of time, and then you can get an idea of the number of organisms that might be there. Now, I'm just going to delete this. Okay. Now, if we look at what the trap does, is you need to have a cover over the top. Now, these work really well on the floor of woodlands, because what you can do is you can put leaves, dead leaves, leaf litter over the top, so it hides the trap from predators as well. So if you are going to keep the organisms alive, then it will keep them safe. Now sometimes at the bottom you can put alcohol in that will kill the organism if you want to kill them, to keep them, or detergent to prevent them from escaping, because some organisms, as you can imagine, might be able to climb up the side and get back out. Now if you use enough of these, and again put them in the right places and randomly sample, then you can get an indication of relative population sizes of them. Now a quadrat is essentially a square. Now if we look at the square here, we have a length here and a length here. Now the lengths of these are 0.5 meters, so half a meter. Now that means that the area inside here is going to be 0.25 meters squared, which is a quarter of a meter squared. Now for example, we could find in this sample 4 daisy plants. Now that would mean that in 1 meter squared, which is 4 times that, we would find 16, as we've got 4 times 4, which will equal 16. Now that would mean in a lot bigger field we'd be able to estimate that there would be 16,000 in 1,000 square meters. Okay, so if we have 4 in 0.25 meters squared, we could estimate that in 4 0.5 meters squared we would have 16. So if we had 1,000 meters squared, we obviously multiply this value by 1,000 and this value by 1,000 to come up with 16,000 in 1,000 meters squared. So obviously using quadrats is a good way of estimating a population size. Now obviously estimating population sizes is very difficult because animals can be very mobile. So to try and prevent this, what you can do is use the capture-recapture technique, which is sometimes called the Lincoln index. Now when you do this, you need to follow a couple of guidelines. Firstly, the animals need to be caught humanely so that they stay alive. The second thing you need to do is you need to mark the animals in some way so that they can be identified. Now that marking needs to be essential that it doesn't harm the animals again. You then release the marked animals back into their habitat. Now you need to leave them for a certain amount of time, then catch a second sample which will contain some animals that have been marked and some animals that have not been marked. Now what you can do then is you can estimate the population size, and that's by using the calculation of population size equals the number of the first sample multiplied by the number in the second sample divided by the number of marked in the second sample. Okay, so I'd like to pause the video in a moment and see if we can work out this problem. So 56 mice were caught, had their tails had their fur removed and then released. Next evening 62 mice were caught. Of these 62, 25 had their tails shaven. Okay, so pause the video and see if we can work it out. Okay, and when you unpause, then we can have a go and work through what the answer is. Okay, so the answer is the population size is equal to population in the first sample, which is 56, times that by 62, then divide that by 25. Now that gives us an answer of 139 mice. Okay, now we need to remember that that is only an estimate. Now with that estimate we have made several assumptions. The first is there is no death, immigration or migration. The second, they were collected without bias, so that didn't mean that we found a known mice home and we stuck a couple of traps outside the home. The sampling was completely random. And the third is that the markings did not affect the survival rate, so it didn't cause any invertebrates to suffocate because of their respiratory problems. It didn't give paint on animals that make them highly visible to predators. Now, zonation is a gradual change in the number of organisms that can occur in a certain direction. So that could be the distance from a road, distance up a beach, position away from a bush in a field. Now, how we can actually measure what these are is by using something called a transect. Now, with a transect, what you do is you lay a measure tape from the beginning to the end, and at certain points, regular points, you take populations of every animal or organism within that particular zone. So you might be doing quadrats and put a quadrat here, then here and here along a beach, and then be looking at what sort of organisms are there as you go along. Now, once you've collected all that data, what you can do is you can actually display that data in the form of a kite diagram. Now, if we have a look at this kite diagram, okay, we've got the distance along the bottom, so it doesn't matter what the distance is, it could be anything. Now, along here we've got the different organisms. Now you can see that we've got two organisms that have a different line. Now what you have is at your first transect here at 0 meters, so this could be on the road, it could be right next to the bush, it could be anything really that you're doing. Okay, now this means that here you had a population this big of grass, whereas here you had no dandelions. Now at the second transect at 5 meters you had this many dandelions and this much grass. And then what you do is, as you go across the distance, you keep counting the number of the different organisms, and you can then plot or draw what looks like a kite, hence the name kite diagram, to show the different number of organisms that you have. Okay, so to review what we've done today, we have looked at how we can actually capture animals so that we're able to sample them. And remember that we need to try and do it as randomly as possible, unless we're doing a transect, in which point it's at various different points on a position away from the road, or from the sea, or from the bush, or from whatever. We've also looked at methods of capturing animals. We've looked at what we can do with the data as well in terms of calculating populations, whether it's using the quadrats or whether it's using the Lincoln index, and then we've looked at how we can display data and look for patterns with the amount of different organisms in a particular area with kite diagrams. Anyway, I hope you've enjoyed it, and I will see you another time. Bye bye.